So first of all, thank you very much for this kind invitation, and I really enjoy talking about this disease, which I knew nothing about 3 months ago. So astronomical learning about this. So my disclaimer statement is I'm a couch scientist. So I've never seen a COVID patient, and I've never they didn't know anything about the disease 3 months ago, and now suddenly I'm like the expert of everything, right? So that's I'll tell you why that is the case. But I read all of these papers. There have been more than 25,000 papers that have been published in the last 3 months. So it's an avalanche of papers that are being published. So it's a phenomenal time to be a scientist, and a phenomenal time to be an immunovirologist, which I am. So which is great. Just a little bit about the task force in India. What we're doing is we meet on a weekly basis, and we look at all the different drugs that are being proposed as potential therapies. We look at drugs, diagnostics, and vaccines, and we review them, and then we score them in various scoring mechanisms, and then we give recommendations to the government what should be the best drug and what clinical trials should be done, and we review the data. So it's done on a very regular basis. There are about 20 people in that task force. So I'm going to talk to you about 4 things. A little bit about pathogenesis. I'll talk about very, very interesting immune response results that are coming out. I'll tell you a little bit about the components of the virus which are necessary to make the vaccine. So let's look at the pathogenesis. So I think most of you will know this, but the key point in the pathogenesis of this pathogenesis is the mechanism by which the virus causes disease. The main thing to remember in this is it's not just the virus. In fact, it may not actually be the virus that causes the disease. It is the inflammatory response to the virus is what causes the disease. So 2 mechanisms are happening at the same time. So the virus enters through the nose, through the or the nasopharyngeal tract. It infects what are called the ACE2 receptors. The virus enters these cells, and it's highly replicative. So it makes millions of copies of itself as soon as it enters the cell, and it disrupts the cell when it replicates. This results in activation of the immune system. By now you know that the virus binds to the ACE receptor. It has another receptor, which I'll talk about later. It binds to these toll receptors also, which are also intracellular. And these toll receptors in an uninfected cell will then activate all these biochemical signals, which are shown here in blobs. Right? One activates the other. It's like a Saathi Haath Banana type, chain reaction. This activates that activates that activates that, and then the signal ultimately reaches the nucleus. The nucleus activates interferons, and interferons go and do these amazing immunological activation functions. The virus has at least 5 or 6 different proteins in itself which blocks the interferon induction pathway. There's NS4A, NS4B, NSP15. NSP stands for non-structural protein. 15, the M protein, the ORF-A protein. ORF stands for open reading frame. 3A and 6A proteins, these bind to and they block the signal transduction cascade, which results in activation of interferon. So the virus is blocking your interferon activation. It's blocking the innate immune system of your body intelligently. So that's the innate immune system. Now, the next is adaptive immune response. When the virus enters the body, it enters the cell called the antigen presenting cell. This cell will activate what is called the CD4 cell, which is one of the main immune systems of the immune cells of the body. These cells are very, very important. They also secrete interferons. When they secrete interferons, they activate another cell type, which is called the cytotoxic T cell. And these cytotoxic T cells then kill virally infected cells like the lung cells of which may be infected inside the virus might be inside the cell. For the virus which is outside the cell, they have to be cleared with what are called antibodies, and you may be familiar with antibodies. These antibodies are secreted by what are called B cells. So we have 2 arms of the immune system. One is called the cell-mediated immune system, and the other is called the humoral immune system. This is a scanning electron microscope of a picture of a CD8 T cell killing a viral infected cell. So the yellow is the virus, is the cell, and it's killing a virally infected cell. This is another picture of it. And this is what a neutralizing antibody will look like. And we'll talk more about antibodies when we talk about the vaccine. The third kind of immunity is called the mucosal immunity, which is present in our parts of our body which are exposed directly to the outside. For example, the nose, the nasal, the pulmonary, where the lung is infected, you have the gut and the genital tract. See, these are our 4 major mucosal tissues, which I will touch upon in the later part of the talk. So that was the immune system. Now let's talk about the virus. The virus has many proteins on its cell surface. I told you there were non-structural proteins earlier. These are the structural proteins of the virus, which basically make up the virus. The spike, envelope, hemagglutinin, the membrane, and the nucleocapsid protein. And it is the RNA virus. This is a very large, this is not an actual picture, but this is a representation of what the viral structure looks like. This is the viral spike protein. This is what the structure looks like. It has a ring-like structure on top with a stalk. And if you look at that ring-like structure and stalk, in a schematic representation, it looks like this. The stalk part of the spike protein is embedded into the membrane. This is called the transmembrane region. And the top part of the portion is this part. And for the virus to enter, this S1, S2 domains of the spike protein have to be cleaved, which means cut, by a proteolytic enzyme, which is present on the cell. And I'll show you a cartoon of that in the next slide. So what happens, the viral entry process requires 3 steps. First, the virus has to bind to the ACE receptor. Then the S1 and S2 have to be cleaved. And then the third is that the virus enters through a fusion mechanism. So I'll show you the cartoon here. The virus here binds, the spike protein has 2 components, S1, which is red, S2, which is purple. And it first comes and binds to the ACE2 receptor. And an enzyme, which is present on the cell membrane, cuts this portion of the spike. And then the virus enters through the other receptor. So it's almost like the first receptor, which is the S1 portion of the virus, is almost like a decoy. It's not the real entry receptor. The real entry receptor is the purple receptor, but it's only exposed after the virus first binds to ACE2. See what happens. Cleaved, enters. So for a vaccine to be effective, the vaccine has to make an antibody not only to the red portion, but also to the purple portion of the virus. And that purple portion of the virus in an uninfected virus, when the virus is not bound to a cell, is not exposed. It is always bound to this domain. So that domain is not available for the antibody to bind. So very difficult to make a virus antibody to this virus. Although a lot of studies have now been done in the last 3 months demonstrating this spike protein, which is this binding portion to the ACE2 receptor, when it binds, it can enter. And when you have antibodies which are directed against this region, they can actually block the viral entry. So there's a proof of principle suggesting antibodies to spike can block entry. This is what the immune response to the overall immune response to the virus looks like. When you get infected, there is a long 2 to 14-day asymptomatic phase when the virus is replicating at a very high rate in your body, but you're not showing any symptoms. These are the asymptomatic people who are infecting other people unknowingly. At some point, the viral load in the body becomes high, and then you start getting symptoms. The immune response starts kicking in. First, you get an IgM antibody response, and then you get an IgG response, which are both antibody responses. And you also get T cell responses, the CD8 T cell that I mentioned. Once all of these immune systems are activated, then the symptoms start coming down. I've put about 30 days for the virus to completely clear. That's sort of what the knowledge is right now. And the goal of the vaccine is to develop both the antibody response, the IgG response, as well as the T cell response. Now we'll move to the vaccine. So the key questions in development of the vaccine are: what is the nature of the protective immune response? You have to have both humoral and cell-mediated immune responses. What part of the virus can make an immune response? And we know from all the work that has been done so far, looks like the antibody to spike may be good enough for now. what kind of vaccine and how frequently will it have to be given to the individual, and how much vaccine needs to be made. In order to do this in the field of pharma, we call this the requirement document as the target product profile, which means this is what we want the vaccine to do. The goal of the vaccine is to prevent infection and also to stop replication once it is inside. The indication is primarily for healthcare workers who are exposed, susceptible individuals, older people, and healthy contacts of people who are infected. The target population can be anything. It could be 10 million, 100 million, it could be 7 billion. Obviously, if we have to start making smaller amounts, then the larger amounts will come. The efficacy will be by demonstrating that you have neutralizing antibodies as well as cytotoxic T cells. There will be a particular dose that will be effective. It should be durable at least for a year, and the vaccine should be stable for at least 6 months, otherwise it can't be shipped very nicely. The process of development of a vaccine usually takes 10 to 15 years, but the world at large is trying to do it in 2 years. So you have these antigens, which then result in immune responses. We know it is spike. This spike protein can be given as a vaccine in all these different methods of making the vaccine. And I'm not going to spend time right now because I want to focus on going to the question and answer session. We develop a process, do toxicity studies, do clinical trials, and you finish the clinical trial. So it will take about 2 years to get at least the first round of the vaccine. There are a lot of companies who are testing the vaccine. I'm going to talk about this vaccine as an example, which uses chimp adenovirus. And I'll explain that to you a little bit more in detail. So chimp adenovirus, which is the University of Oxford in England, has been developing this vaccine in collaboration with Serum Institute in India and AstraZeneca for the worldwide distribution. It is an adenovirus, which is the virus that causes the common cold. What's done from a genetically engineering perspective, the viral genes, there are certain viral genes that are removed from the adenovirus and replaced with a gene for the spike protein of COVID-19. And this kind of vaccine has been proven to be successful to induce humoral and cell-mediated immunities for other pathogens in the past. There's no approved vaccine of this, but there have been some trials that have been done. So now the results of the animal trials have already started coming in. So the first animal study trials have come in, and this is what the results look like. So there are a group of rhesus monkeys that were given, 6 monkeys were given the COVID vaccine, and 3 of them were given a control vaccine, which they were given the adenovirus expressing green fluorescent protein, which should not induce COVID-specific immune responses. It was given intramuscularly. You measure the immune responses, and 28 days later, these monkeys were challenged with the live COVID-2 virus. So first, let's look at the immune response. The red dots are the monkeys who got the COVID vaccine, adenovirus expressing the spike protein. And as expected, the animals who got the vaccine developed a virus neutralizing titer, which is quite high and good. They also developed T cell responses, like I showed you, cytotoxic T cell responses measured by what are called spot forming units per million peripheral blood mononuclear cells. So that's what this readout is. The readout is, this is T cell, this is the antibody response. Successful so far. Now, before we go into the challenge, let's look at the respiratory system of humans. We have what is called the upper respiratory tract and the lower respiratory tract. And this becomes important when we look at the result of the vaccine. The upper respiratory tract comprises of the nasal cavity, the oral pharynx, etc. Right? And below the red line, approximately, is the lower respiratory tract, which includes the lung and the deep lung. A nasal swab is taken to understand what the amount of virus is there on the upper respiratory tract, and a bronchoalveolar lavage, which is BAL, is taken where you put a tube through the trachea, and then you collect a sample from somewhere around deep here, and that is your bronchoalveolar lavage fluid. A little busy slide, so I'm going to just put a box around the middle part. Let's look at the middle part first. And what we're doing is we're comparing the amount of virus that you can detect after the monkeys have been challenged with the live virus. You can see that the red dots are lower than the blue dots, which is a good sign, which means in the lower lung, vaccinated monkeys have much less virus than the non-vaccinated monkeys. So the vaccine is working in the direction we want to work, right? Now let's look on the left side, and here we are looking at the clinical score. Again, the red is the vaccinated monkeys and the blue are the control vaccinated monkeys. You can see that the vaccination doesn't prevent the virus from entering from infecting the body, because even if you're vaccinated, even the vaccinated monkeys have a large amount of viral replication, but they clear the virus in 5 to 6 days, versus the control monkeys, which control it, take a little bit longer to control. So therefore, what is called sterilizing immunity is not induced by this vaccine. At least this data suggests that. If you look at the nasal swab, there's no difference between the red and the blue dots. So the vaccine, when you get the vaccine intramuscularly, and you generate an antibody response, which has been demonstrated, that antibody response is not high enough in the mucosal tissue that you will not, even if you're vaccinated, you will still get infected. But if this holds true, but you will get a lesser form of the disease. That's what this vaccine may do at this point. This is the first vaccine. There are 100 other vaccines that are in development. So the summary of this vaccination study is it does not induce a sterilizing antibody response. It induces both cell-mediated and humoral immune response. It enables the clearance of the virus in the lower lung, but maybe slightly less effective in the upper lung. And it prevents pneumonia, which is the main thing for this disease. You want to stop the progression of the disease to pneumonia and death. You have to make this vaccine in very, very large scale. I'll skip this slide. If there's any questions, I'll go through it later. And this is the design of the clinical trial. About 1,000 subjects are being tested in the clinical trial. Half of them will get the COVID vaccine, and half of them will get the meningococcal vaccine, which will not protect against the COVID vaccine. And then after a predefined number of individuals have been infected, the study will be unblinded. So right now, everybody is getting a vaccine. They don't know who's getting what. And when we get 100 subjects who are infected, then we look at the results. So let's say the study is completed. It's not completed, but this is the potential result that could happen. If these 100 people are infected, when we identify them, one possible result could be all of the 100 infected individuals were in the meningococcal arm and none in the COVID arm, which means the efficacy of the vaccine is 100%. 80 of them could be in another probability is that 80 of them would be in the meningococcal arm and 20 of them could be in the COVID arm. That means the vaccine is 80% successful, et cetera, et cetera. So there's a possibility. There's also a possibility that there's no difference between the number of people getting infected in the meningococcal and the COVID arm. That would be 50-50 individuals, and that means the vaccine would fail. There's actually another possibility. In fact, the vaccine may have a larger number of people infected than the control, and your result would be minus 20%. And in this situation, there's a possibility that your vaccine could make the person more susceptible than if they didn't get the vaccine. So why am I saying this? Because there's a theoretical possibility that this can happen, because it has happened for other vaccines like dengue. So finally, the criteria of a successful vaccine should be that it should be efficacious, it should induce mucosal immune response, it should be safe, it should have economic, social, and political impact in society. And so I'll quickly summarize of what I talked about now, and then we jump to the questions. I talked to you about the components of the virus, the immune response. The virus is actually attacking the immune response. I think I mentioned that to you, especially the innate immune response. And I showed you a little bit about the vaccine. So let me stop sharing and pause a little bit before I jump to the slides. Okay. So Janavi, I'll come back and see if there are first any live questions that come up. Otherwise, I have a whole slide deck of about 10 or 15 questions that you have sent to me. I have responses to all of them.
Wonderful. But this was also incredibly good to see, right? Because I was just thinking it is so complex. I think one of the dangerous parts of is that we have a tendency to maybe simplify things, right? Even in terms of like the precautions we're taking or the seriousness of the virus, there's a lot of simplification happening. And so I think maybe people don't understand how difficult it is to make a vaccine, right? Or to address this pandemic. So thank you. That was lovely to see. We did have some questions during your presentation. So let me start with the first one. So Arunima asks, the fact that the virus can block interferon activation. So does that mean that the virus has mutated to reach the stage, or was it always the smart?
Smart, I don't know, smart or not, but it's trial and error. So first of all, the virus is about 300 million years older than humans. We are only about 2 million years old. The viruses have 300 million years old. They're at least, if not more, right? So they are much more evolved than we can ever be from their perspective, right? So, but the mutations that occur, like there are almost every virus that we know, whether it's HIV, hepatitis B, HPV, every virus that we know attacks the immune system in a very particular way. It's a war that's going on between the immune system and the virus. And when the immune system attacks, the virus tries to mutate away from that attack mechanism of action and tries to try something else, right? So this mutation has been not occurring now. It has been occurring for the last millions of years. We are just able to understand that now because of the technology that we have. That LSP15 binds to ORF3A, that we know now. The virus knew that 300 million years ago, when we didn't even have names for these proteins. So it's mutating, but it is not that this family of viruses doesn't mutate as much as, let's say, influenza or HIV. It doesn't mutate that much.
Okay. We have a question. We have another question. So I'll just read it out. We've heard that in some people, their immune system's response to the coronavirus causes clots and strokes. Is there any more insight on this? Why it happens? And need to prevent it?
Yeah, so that's a good question. I don't have a direct answer on that, but I think there are now more and more reports coming out that there is a mechanism of action where the virus may be interacting with the clotting system and causing these thrombosis events, which then ultimately lead to clots and cardiac arrest or even strokes. The precise mechanism of this, I don't have it right now with me, but essentially it impacts the clotting cascade.
Okay. Moving on to another question from Padmini again. So her question is, a lot of people are advising that we should buy, a pulse oximeter to keep a check on oxygen levels. So especially if you're showing symptoms. And so of course, these are now flying off the shelves. So is it something that you should invest in?
Well, I'm not a clinician, so I don't know the clinical manifestation of this, but I have a slide, which later on, Jahnavi, I'll send you that slide deck that you can share with anyone. All the questions that you have, I've got a systematic answer to that. And there was a recent WhatsApp video. I usually don't pay attention to a lot of WhatsApp videos. I like to go to the source. But there was one that by Sandhya Ramanathan from Auckland. I don't know if you've seen that video of hers. I have it in the slide. If you look at her video, she's a physician and she's COVID's patients, and she recommends an oximeter if you have it. And by measuring the amount of oxygen that is in your body, you may be able to track how bad your disease is if you've got the infection.
Okay. Great. So we have a few more questions that are coming here live. The first one is on herd immunity, which is, given the challenges with vaccine development, can you explain a bit about herd immunity?
Okay, so this is a good time for me to jump into my slide, because if you can enable my slides.
Yes.
Because it's easier for me to talk to you about with the slide that I already have.
Right.
I think somebody has to allow me to share my slides.
Yeah. I think you should be able to.
All right. I have a picture which is 1,000 words. So I'll show you that slide deck. This is my last slide from my previous presentation. And I have made a whole list of all the questions. These are all the answers to your questions that I have. Great. So let me see, where's the herd immunity question? The herd immunity response is here. Okay. So what happens in herd immunity? What is herd immunity and how do we build it? So the principle of herd immunity is that if there are enough number of people infected and have antibodies, then you will prevent spread of the virus, right? So this slide sort of depicts that process. Green are the uninfected people, in the population. Black dots are the previously infected people who have cleared the virus and now they have antibodies, and so they won't get infected again, right? Red are the uninfected, are the infected people who are walking around in society, maybe asymptomatic, but they might be walking around in society. And the red in the box are the infected people who are hospitalized or sequestered at home, or they're very sick. So they are not infecting anyone at this time. But in the population, these are the people who are walking around in the population. When the vaccine comes along, they will be the blue dot ones. But of course, we don't have the vaccine right now. But if you consider let's first look at when they have the vaccine. If you have enough people vaccinated and enough people infected, then let's look at the scenario between in this space. There's a person who's infected. This person will be only be able to infect an uninfected person, that this green dot will become red. Right? Once these two people are infected, they cannot infect anyone around them because they're either previously infected or vaccinated. So then the virus dies off in this population. That is the principle of herd immunity. Now the question in a larger population, and there are a lot of studies being done right now and modeling studies that are being done, the range of people that need to have either vaccinated or have been previously infected ranges from somewhere between 30 to 80% of the people have to get infected before you get herd immunity. I mean, we don't know the exact number. People are trying to model it and try to figure out. Worst case scenario, 80% of the people have to get infected before you get protection. Best case, 30%, but I think the number may be somewhere in between. So that is the principle of herd immunity. If there's a follow-up, I can answer.
I think that, I have a follow-up, and I think it's very similar to a question that somebody else has asked, is how will herd immunity get affected, especially if some previously affected patients are showing symptoms again?
Okay, so that is a question related to, do you get infected again?
Someone had asked it earlier, and I can see it in the Q&A as well, is if coronavirus patients can get reinfected after full recovery.
Right. So the question that you had sent, and I think this is the same question, why do some people who have previously got COVID get it again? Right? Mm-hmm. So we don't know that for sure that people are actually getting infected or not. These are anecdotal reports. Mm-hmm. There are, what is today's number? It must be 7.5 million people infected in the world, right? And the reports of people getting uninfected are less than 100 or 1,000. So the number of people who are getting reinfected, the reports that are coming out that people who are getting reinfected are a very, 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 very minute percent of people. But the news about them is massive because the news is alarmist, right? So I'll tell you what the these are 5 potential reasons why we think that people may be reinfected, right? So these are, for example, first one can be people hospitalized with COVID. When they enter, they test negative because the assay doesn't work for some reason. The assay wasn't good enough. Then they get discharged as negative. Mm-hmm. And when they go home, it comes up positive. So then, somebody may say, aha, they were negative before, now they get become positive, so they got reinfected. But that was not because of that, because the test was not good enough, right? The second reason could be the first infection resulted in low level of neutralizing antibody, right? The antibody response that was generated against the first infection, well, the antibody response was not strong enough. So you actually got infected again. That could be one possibility. Then another possibility is during the shedding, and if you're looking at a person who is asymptomatic and the person goes 2-3 times to get tested, because again of the inaccuracy of the test, sometimes the patient comes up positive, sometimes comes up negative, sometimes again goes again, comes up positive, negative. So then the interpretation of that, I was negative before, now I became positive. So the interpretation could be not because of what's happening with the patient, but because of the kit not being sensitive enough. And that is the fourth point, which is the low sensitivity of these tests that measure the virus are creating this whole situation where they actually may not actually be reinfected, but but there might be all these other anomalies. And the fifth reason is the actual reason that we should be worried about, is there actually may be a mutation in the virus that actually results in reinfection, right? There is a theoretical possibility of that, and people are modeling to see what is the possibility that could happen. There are 2 or 3 reasons why I don't think that that is happening. One is historically, mainly 300 million years history suggests that this family of viruses doesn't mutate very much. It does mutate, but not at the rate of what HIV and influenza does. So the likelihood of the mutation will be formed that will suddenly start infecting in a different way than using what is the worst case scenario? You have a mutation that infects you, which is not using the ACE receptor, using some other receptor to infect you. That is not happening, right? And the other thing about the whole purpose of the whole philosophy of vaccination is if you get vaccinated by something that is similar to the original virus, that is what you make a vaccine against. That's how Jenner found smallpox. He used the cowpox to make a smallpox vaccine. So in fact, if you get vaccinated infected by a particular strain of virus, and you get infected by another strain, you should actually have less pathogenicity of the second virus. So hopefully that answers the question quite detailed.
No, that was very helpful. Yeah, that makes sense. I don't think that's been talked about enough, right? So, and of course, then you're also right about the fact that it's a very small number. We don't really have enough information about these patients who previously got COVID getting it again. But it's still, I think, something that's playing on people's minds.
Yeah. Especially
since a lot of people are recovering nowadays and are coming back into society, right? From hospitals and things like that. So yeah. So difference between a heart attack triggered by COVID-19 versus just a blockage of arteries if that patient is not tested for COVID-19?
So you broke up a little bit, but the question was, are you getting a heart attack because of COVID-19 or is it just something else?
Yeah, is it just a blockage of the arteries because the patient has not been tested for COVID?
Yeah, I think, again, I'm not a physician and I'm not going to give you a clinical answer to that. I'll give you a common sense answer to that. So if you think you're having a heart attack, you shouldn't worry about whether it came from COVID. That's not the time of thinking of the mechanism of action of how you got the heart attack. Take care of the heart attack first. Then worry about how you got it. If you survive, then you figure out how you got it. Just whether it is induced by COVID or whether you ate too much fatty food or whatever, don't worry about how you got the heart attack. Please take care of the heart attack first, right? The mechanism will come later. So, and what I will tell you, that was sort of a little bit arrogant answer, I know, but the reality is that since the heart does express ACE receptors, there is a high likelihood that if you're infected, that the probability of having cardiomyopathy, which is sort of heart-related problems, is significantly increased. And that's why older people get more pathologies than younger people. Yeah?
Right.
Okay.
Right.
Good. Very good. Very thoughtful question, though. Yeah. Thank you.
So now, Naren, do you want to maybe walk through some of the questions that have been asked previously, and we can see your answers and what you said about them?
I'm dying to tell you the answers to the questions that I can come up with that you asked, because some of the questions were just fantastic. I had to really think about some of these questions, and I don't even know that I've got the right answers yet. So this is a list of all the questions. I've taken each question and tried to answer it systematically. So the first question, first series of questions that you asked, was related to the virus, on the virus. So what makes COVID so dangerous and what makes it spread so rapidly? So what we know right now from the genome, which is the RNA sequence of this virus, that it is quite similar to the SARS-CoV-1 and the MERS virus that came about a couple of years ago, right? But this virus is 3 times more infections than the previous one. That's what the sequence analysis suggests. The transmission is not airborne, but is almost airborne. It goes through these droplets. And the third is that it causes asymptomatic infection when transmission occurs in unsuspecting ways, right? So you may be infected, you're asymptomatic, and you're walking around, and that'll cause infection. And Atul Gawande wrote a very nice piece in the New Yorker, this is the picture from that paper. So while we focus on the 6 feet, which is the predominant infection that you see, that's when you're standing near, if you're infected, highly infected, that can go to 9 feet, 12 feet.
Wow, that's a very impactful image.
Yeah. So that's why it spreads so dangerously. That is this question. Then from a structural perspective, I told you that this SARS and MERS virus are so we know from a sequence analysis that the spike protein of SARS-2 has a specific insertion of nucleotides in the spike protein that are hypothesized to make it more infectious. The red portions of the spike protein that you see are mutations which are specifically present only in COVID-2. They're not present in the earlier viruses. And therefore, there's an extent to the extension of that is those are the sequence changes that are making the virus more infectious. That's a paper that was published in Molecular Cell literally a couple of months ago. That is the second question. The third question is, what is your view on how SARS spreads so rapidly in a country like US, wherein the population density is low and merely a quarter of that in India, right? So I think this is the picture that I wanted to show of what is happening if you don't have social distancing. If you don't have social distancing, every red dot will make every green dot red. Like you'll get rampant infection no matter what you do, right? And that's why social distancing becomes important. And that's what we've been doing in India for the last several months. And that's why the number is low. So now, what are the factors in India that could potentially, or anywhere, not just in India, what are the factors that contributes to the rapid spread? First, let's look at the physical factors. The number of people that were originally infected in the country. So when you look at India, in February, or was it March, maybe March, there were estimated about 500 people came to India who were infected. And they came primarily from Europe, not necessarily directly from China, right? And so 500 people infected, whatever, a billion people in India, so to speak, right? What happened in the US? From December, January, February, March, 4 months, right? Every day, 3 million people from China went to US. Every day for 4 months, right? And these are the uninfected people. So a lot of virus has been put into the US. Of course, you'll get much more rapid infection. So it makes absolute common sense that you will see in California, probably 1 million people came for 1 month directly from Wuhan itself. Unknowingly, right? They didn't know that they had the infection, they just came. But they brought the virus with them. The second physical factor, that the first one is how the number of people originally infected in the country. The second is the density. What is the density? And we know about this. The third is the intensity of distancing, which India has been very good at. And the fourth is the physical use of masks. There may be many other physical factors, but these are ones I thought about immediately. Then there could be viral factors, that the sequence of the virus that is infecting India versus what is infecting US could be different. There is some data to suggest that it might be different, but I don't know that data myself, so I'm not going to comment. But right now, it seems like that is not a major factor. It could be, but I don't think it is. But we'll see what the data shows later. And the final is the host factor. Are people in India less susceptible to the virus than the rest of the world? So do we have more immune strength? Do we have less comorbidities, and is age an advantage? And to answer to all of that, the answer in India is yes. And so I'll show you this. This was supposed to be a picture. Let me get this read. I wanted to show you this thing. It was animated. So let me show you this. This picture, this modeling study was done. Actually, this modeling study was published in March, when we had the first lockdown. And what the model was, I think this is March 18th or something. It's very tiny, but the first one is March 18th. And if after 21 days, if you had unlocked, it would have gone tremendously, it would have gone high, right? Now, if you did a second lockdown, then it will still go high. At that time, we thought if we had a third lockdown, it would all go down. But these models are not playing out, right? We are seeing that even after 3, or how many lockdowns do we have? 5 now or 4, right? Even after that, you're seeing a rebound.
Right.
So all of these factors are playing into the rebound. So, I'll just put it in perspective. How many people are infected in India now? 300,000 or something? What is the number?
Yeah, I believe it's something like that.
300,000. In the US, I think we are close to 2 million already.
Yeah. Almost 400,000.
Almost 400,000, right? So just to put it in perspective, influenza, which is influenza 1918, which is less infectious than this virus, infected 500 million people in the world. 500 million people were infected with influenza, 1918 influenza, and 50 million people died. And that virus is less infectious than this. So we are nowhere near, if it plays out like the way influenza plays out, we are nowhere near. We are what, 8 million in the world. That was 500 million, and 500 million when there was no travel in 1918. And 500 million, at that time, the world population was somewhere like 2-3 billion. Right. Now we have 7 billion. We travel like hell. So, I hope that doesn't happen. But if I just put 2 and 2 together, I think we've not seen the end of this. So that's sort of not great news about this. We'll have to learn to live with this virus. And what do we do? And I have a slide on that, and that WhatsApp that I showed you was very informative for me. Okay, the next question is, why are certain people more susceptible than others? So there are many hypotheses. Why is the immune system of people who live in places that are less hygienic better? There are several hypotheses. One is we might have a heightened overall innate immune response. I told you that the virus attacks the immune system, right? It attacks the innate immune response. But if we are exposed, what happens to any exposure? Our innate immune response is higher. The innate immune response that we get from any virus, whether it's the BCG vaccine or whether it is tuberculosis or whatever, all the dirt and filth and everything that is around us, and everything that is actually saving us from these infectious viruses. That increases our baseline of immunity, and that might be one of the mechanisms, by immunological mechanisms, why we are not getting progressive disease. That's one hypothesis. Second is, if you have a higher baseline, the level of virus that will grow in you will be much lower, and therefore your immune response will not be exhausted. What happens when you have a very high viral load, when the war is going on and the viral number is very high, your immune response also has to fight at that level. And literally, I'm using the word exhausted like it. Here the word is actually, there's a biochemical understanding of what an exhausted T cell looks like, but it also could mean the English language word. The third is that the presence of high titer antibodies to RBD spike, which is the receptor binding domain spike. So if you have a higher innate immune response, if you have a very strong immune response, you might generate a higher titer antibody, and therefore you might get better protection. The fourth could be the rapid expression of what are called checkpoint inhibitors. The checkpoint inhibitors are these proteins in the immune system that are responsible for shutting down the system. So I told you the pathology of the virus is twofold, right? One is the virus induces pathology. The second is the immune system induces pathology. So we might have a better shutting off of the immune system so that immune-mediated pathology is not seen in people who have higher infections. And then what has been recently shown is that a certain kind of gut microbiome, like certain kind of bacteria that are present in the gut, actually may be able to develop an immune system that is protective in nature. All of these hypotheses are being tested formally in different experiments. And all of these are not just casual statements. There are like 10, 20 papers on each one of these topics.
So is it a fair assumption to say that the roadside chart that we are eating is helping build our immune response?
It for sure is. But you might die from the typhoid that you get also.
But it's helping with immunity for something else.
The English statement, what does it say? If it doesn't kill you, it'll make you stronger. That exactly applies here. That roadside chart may kill you, but if you survive, then you'll be stronger. All right, let me move to the next question. I'll pause here because I can go on. There are many questions I'm answering, but if you want to ask a live question, I'll be happy to answer that question. I know Janavi is off, so Anjali, you want to see if anybody has any questions?
Sure. So let's see what's the question.
I mean, I have answers to all the questions that you've got like this in a detailed way. So if you want me to continue, I can do that too.
Yeah, sure. Or maybe we can take one or two questions which are live. So Manjri asked a question. She's saying, given the challenges of vaccine development, could you explain a bit more about herd immunity? But you already did that. So coronavirus reinfection, you already answered. So maybe Shri Ramya asked a question about if the COVID-19 virus undergoes mutation, then what will be the next steps? How will be overcoming that? But you already answered that question also. Do you want to add anything more to that?
No, I think as we get data, if there's more mutations occurring, we will have to make vaccines against that mutated virus. For example, today's influenza vaccine, or let's say hepatitis B vaccine, or even human papillomavirus vaccine, you get 9 different virus vaccines you get when you get one single vaccine. When you get a papillomavirus vaccine, you get 9 different viral vaccines in one. You don't know that it's 9, but there are 9 viruses. When you get the pneumococcal vaccine, it has 23 different strains of bacteria in that one vaccine.
Okay, sure.
We don't know that, right? But there are, your pneumococcal vaccine that you get has 23 different strains of pneumococcal bacteria.
Interesting. Yeah, we just think that we are just getting a one shot, but it's a combination of many things. Yeah.
Yeah, it's a combination of many.
Yeah. So there's one more question from someone. He's saying, is there any evidence for the lab release theory? Many of the friends are believing in that. What's the theory? So
origin of the virus.
Origin of the virus. Do you want to talk about it a little bit?
I mean, I think you can think about conspiracy theories forever, and all great things. I'm sure there is a James Bond movie that's already being designed to make this conspiracy theory financially viable. But I'll tell you what my perspective from reading of the literature is. I mean, I'm going to assume that people are good in general. I don't think people will be that malicious to make something like this, which is to take over the world. First of all, that is one. The second, and you may say, you're being too naive, right? There may be people. Then I'll say on the same side, those people are definitely not that intelligent that they'll make such a fantastic virus that will destroy the human race. Humans are not that intelligent that I'll think of making something that will destroy the human race. Oh my God, that human doesn't exist today. Yeah. So, but from a sequence, the jokes apart, when you look at the sequence analysis that I showed you, those three or four sequences that were responsible for that occurred in the spike protein, right? Mm-hmm. And those are the only differences between COVID-1 and COVID-2. So if someone actually maliciously did that, right, they have to be either super brilliant scientists to know that, okay, that sequence is going to change my infectivity by 3-fold, and therefore I'm going to release it into the Wuhan market and do all of that. That seems unlikely to me.
Yeah. I'm on your side on that. Yeah, I saw a lot of movies are moving around, a lot of viral videos are going. Maybe it's a newsworthy content. Yeah. So
but make a movie of it. Go make a movie of it.
Yeah, sure. Make a movie, watch on Netflix. Yeah. Okay, so let's take a question from Aarti. RT saying, how could one understand the difference between the heart? I mean, you already answered that too. How could you understand that? Because what's happening, this is happening a lot in India. People are getting strokes. So sometimes people are not getting whether it was a heart attack or it was the result of some kind of thing that happened by blockage of arteries because of COVID. And this is something which is happening a lot. So a lot of deaths are happening and people are not sure about it. I think that's where she's coming from. If you can give some.
And I guess the compassionate answer rather than the aggressive answer that I gave earlier was, I think it'll be very hard to decipher whether if you have COVID infection and you get heart trouble, right? The likelihood that COVID is causing heart disease is very high. Very high. So, if you get COVID infection and you are a heart patient, please contact your cardiologist tomorrow or like today if you are infected. Because, it's actually pretty simple to understand, right? The heart cells contain the ACE receptor. It's like darwaza hai, khula hai.
Yeah.
There's no reason why it can't enter. It just happily enters. There's nothing to stop it.
Yeah.
And if your heart function is already compromised by, let's say, 50-60%, a little bit more compromise, and then the virus, and then your heart will stop functioning. So it is not very difficult to understand from a biology perspective.
Sure. So you are saying rather than anticipating, if you are a heart patient or if you have somebody in the family who's a heart patient, and if you think they have mild symptoms of COVID, contact your doctor ASAP.
Yeah.
Sure.
This actually may be a good time if you let me to share the video. I'm not going to show you the video, but I think this lady, this doctor, just yesterday or day before gave a very nice WhatsApp video. I would highly recommend it. I'll send it to Jahnavi, the actual video, and she can share it with whoever wants. Her name is, I just took it from the WhatsApp. And this is not all the detail. Don't go on my slide. Listen to her. It is on YouTube. Her name is Dr. Sandhya Ramanathan. She's from Auckland, New Zealand. She's very, very nicely described what you must do to, if you think that you're going to get infected or you got infected, these are the things you must do. She talks about priming the immune response, how to reduce the viral load, gargling steam, doing breathing exercises, take a pulse oximeter and monitor the oxygen in your level. And she even tells you how to sleep if you have infection. So she tells many more things. It's almost like a 20-25 minute video. So I would highly recommend looking at this video if you think that you got infected.
Yeah, sure. So I mean, we can share the video with all the attendees as well. I think
I'll send you the slides also, to all the slides, and this slide I'll send it to Jahnavi and she can share it with whoever wants it.
Okay, lovely. Yeah, so Jahnavi, to you. Yeah.
Thanks, Anjali. So sorry about that. I think just a question that I was thinking of, I think someone had asked earlier, based on just heard right now about people who are susceptible to it, right? So someone had asked in the questions was, how do you take care of seniors with immunocompromised systems, especially if their caretaker has to go out for work or has to grocery shopping, things like that. So what can you do in that case?
Sorry, what were you saying? Caretaker is sick, or what would you say?
No. So what can you do if you're taking care of you as a caretaker, you're taking care of senior citizens, right? Who are probably have immunocompromised systems. Right now, you as a caretaker have to do other things as well. You're going to work, you have to go grocery shopping, things like that, right? So you are getting out of the house, so you can't really isolate yourself. Yet you're living you're living with people who might be possibly immunocompromised or who are senior in age, right? And I think that's very true for a lot of people here who are living with their parents, grandparents, things like that.
So what would you do, right? So I think first of all, listening to her would be very, very useful. And I think using your mask and trying to maintain social distance as much as possible would be very useful. Remember, I mean, that this is a very, very dramatic video, right? This is from that. I'll show you that this is. Remember that this is happening. Consider that if you go out and come back, and you come back to your grandmother, consider that you're infected.
Mm.
And how would you stop this? Don't go near her or him, and wear a mask. Wear wear a triple mask if you have to. If you've gone outside, and then every time you go outside and every time you think that you're infected, this is what's happening to you.
Right.
You don't know it, but this is what you're doing. That these droplets that are going parallel are just when you're talking.
Right. Right.
So remember this video, like ingrain it into your brain that this is happening if you're infected. And whoever comes near you, that's the level of infection you will get.
I think that's, I mean, it's difficult to implement, right? Now you're living, now grandparents, you have maybe you have a layer in between your grandparents and yourself, because you have your parents, right? Directly in communication with your grandparents. So you're not directly taking care of your grandparents. What if you're living with elderly parents, right? And you are their caretaker, or you have kids, you have babies, you have a toddler. Someone else had mentioned that as well, right?
Yeah.
What can you do in that case?
Try to be, you know that the route of transmission is through the oral mucosal tissue, right? So if you think that you're infected, you know whether you've gone out. If you sit at home for 14 days and you've not gone out anywhere, at this point, you can hug your grandmother and you can kiss her and you can do everything that you want. If you've not gone out for 14 days and you've not been exposed to anything, then you're not infected. Then it doesn't matter, right? But if you've gone to the grocery store and you've gone to a crowded local, or you've gone somewhere like to drink coffee with a lot of people and you don't know whether they're infected or not, right? And one of them has this kind of, one of them looks like the picture at the bottom that I'm showing you here in the slide, right? And the virus has entered you. If the virus enters you, there is absolutely no way that you can prevent the virus from replicating. You may not get symptoms, but that virus will replicate in you, and then you will be able to transmit to someone else. So that's the thought process that you must have.
Right.
Life has changed. You have to wear. It's almost you have to wear a mask all the time at home. I mean, I think just trying to use common sense of the pictures and the things that I've shown you here, and applying to your daily life. Just think about this picture. Like I'm going to show it again because the impact factor of this picture is massive. I love this picture, right? This is if you're asymptomatic. This is if you're symptomatic. If you have a lot of, the higher the amount of virus that's in you, the further you will transmit.
Right. Exactly.
And how do you prevent this? You have to use your common sense to see how you can prevent this.
Right.
There's no instructions I can give you for you to figure out these things.
Right. No, that is very helpful, because I think, again, the fact that there's low awareness, right? Because there's been so much oversimplification of both what causes it and how you can take care of yourself and the precautions and things like that, right? I think it might be making people almost more comfortable, right? And it might lead to people not taking care of themselves. So I think that's really important for people to see.
Yeah.
Right?
So I would say definitely listen to her, because if you're going to get infected, right? And if you're living in a populated place and you're going to be exposed to a lot of people, chances are that you will get infected sooner or later. It's not a matter of if, it's a matter of when, right? You can't not have this virus. You're going to have to live with this virus. And these are the good things that you have to do. You have to take care of your whole. This is a good time to take care of your health. The healthier you are, the better off you are with this virus.
So I think you're still in the middle of your slides, right? So do you want to go back to them now?
Yeah, I can continue.
Yeah.
So I think we covered this, right? We covered this. We covered this, right? In fact, why are people less susceptible in India? Why are certain people more susceptible than others? This is the opposite of what I said. So I made the slide, so might as well look at it. Why are certain people more susceptible than others, right? We know approximately 80, maybe even 90% or more of the people who are infected are silent killers and are asymptomatic. That's what we know, right? So the question is, how are they asymptomatic? Meaning, Why are they asymptomatic when they get the virus? I just mentioned to you that they might have a higher innate immune response. And because they have a better immune system, they might be able to clear the virus faster before it causes a lot of pathology. And lower viral load means less morbidity and mortality. The more the virus, the worse it is, right? Who are the ones who are more susceptible? The younger and healthier are less susceptible. Patients with comorbidities of lung, heart, gut, liver, any comorbidity that you have will make you more susceptible, because that organ will dysfunction because the virus infects. And why are certain people more infection than the others? That the ACE2 receptors are widely expressed in many tissues. In comorbidities, their organs are performing suboptimally. That's why they have the disease. And also other mechanisms of why people have differential infections include, I told you a little bit about, it seems this is new research that's coming out, that depending on the nature of the gut bacteria that you have, you might have what are called pathogenic bacteria for this disease or protective bacteria for this disease. So, but that research is coming out. The preliminary data suggests that there seems to be a classification of these bacteria. Not to jump to any conclusion, but every person has about 1 million different types of bacteria in your gut. So there's no easy way to say, oh, mujhe wo bacteria de to main bach jaunga. So it's not that easy. And then of course, there is some genetic predisposition. If you express certain genes, like immune response genes or something like a genetic component of yourself that might be protective against this virus. There is some early data that is coming out, but nothing conclusive yet. So these are the various reasons. The next question is, why do some people who got previously infected get infected again? I think I answered this question, but let's go through this systematically. We know what happens when you get infected the first time. We know that the immune system generates antibodies. These antibodies are made against several parts of the virus, including spike and everything else. These antibodies bind to the ACE receptor binding domain of the virus. These antibodies block the virus from entering the ACE2 expressing cells, and therefore they're called neutralizing antibodies, right? There are some infections there are many infections that you get. Once you get them, you never get them again. Like varicella, which is chickenpox, measles, smallpox. Once you get these infections, you'll never get them again. But then there are other infections that you keep getting again and again. And one of them is influenza. Every year you have to get an influenza vaccine, and every year you have to. And we think that we're getting reinfected, but actually we're not getting reinfected. This is what's happening. Remember, I showed you that there are 3 steps to enter. The virus has to first bind to the original receptor. It has to cleave, and then it enters through another receptor. This is the mechanism of how the virus enters the cell. Okay? Now, neutralizing antibodies need to be directed against both the binding as well as the fusion domain of the virus. It has been extremely difficult to make an antibody for the fusion domain of the influenza virus. Hence, even today, we don't have a universal influenza vaccine. Influenza also uses the same mechanism of entry. And we don't have an universal influenza vaccine yet. So it's been very difficult. So this process of making a vaccine against this vaccine is very difficult. And this is the influenza virus. It reinfects every year. It's not the same virus that infects every year. Every year it's a different strain. So in 2016, it was California, H1N1, PDM1, Hong Kong strain, Brisbane. These are all the names of the different viruses, influenza viruses that are exposed every year. This is 2020, 21, Guangdong. These are there's a nomenclature. So every year there's a different strain of influenza that comes, right? This is the sequence variability of the coronavirus that is occurring. And you can see that whatever sequence that is occurring in those regions have a predominant, that particular sequence is predominant. But if you look carefully, while they're looking like they're different colors and therefore they might be different viruses, they're actually not very different from each other. Only very minor differences between each other. This is coronavirus. And then why, this I answered this question, right? Why do people get previously, I went through this before, we went through herd immunity.
Right.
Do you think that the lockdown in Maharashtra was good enough? And why do you think there are still cases increasing despite the lockdown? What I'll say is the virus is highly infectious. I think you already got that, right? And if you're going to be infected, if this is the population and you're infected and you come out into the population, you are going to infect other people. There's like no two ways about it. If you come across a person who's infected and that person sort of so much as speaks to you and that person has virus, you will be infected. Now you may or may not get symptoms. Okay, then we talked about precautions, using a mask. I think from a precaution perspective, I really like her. I'm again going to tell you to see her video. It's really nice. I really like it. And then finally, the questions on vaccine. I think I answered some of these questions during the presentation. There was one question I actually have no answer to. I don't even know about this. What are the treatments currently available for G6PD deficient patients? And I would say go to your doctor. I really don't even know about this disease. So I think that's it. Those are all the questions that you guys had. And any more questions?
Wow, that was great, Naveen. Thank you so much. I don't see any more questions over here, but I did have one, which was just a couple of follow-up questions to something that I saw on the slides. Chickenpox does happen again, right? It occurs as shingles in adults, right? And so that I was just wondering, because you said it doesn't really happen again, is that like a mutation again, or what is that?
So
is it a different virus?
Yeah, no, it's the same virus. I'll tell you what happens with chicken. What chickenpox and shingles is. Chickenpox is called by varicella. Varicella is the name of the virus, and when you get infected as a baby or children, then you, just like this virus, you actually inhale it, and then you get infected. And then what the virus does, the virus replicates, and unlike this virus which infects the lung, varicella infects the nervous system. It infects all your nerves, and it grows in the nervous system. But most of us have a very strong immune response, and so therefore we get rid of the virus. The viral particles and all the virus antibodies and everything are cleared. But there may be one viral RNA, one viral DNA particle that might be sitting in one nerve in your body. Because it's not a virus; it's just the DNA of the virus that is sitting in your nerve of your body, right? It's still, the virus genome is still there. So what happens is, in next almost 30, 40, 50 years sometimes, your immune system is not doing anything. Your immune system is very strong. So as you grow older, when you get some kind of an immune suppression, your immune system, sort of the threshold of your immune system goes down, that DNA that is sitting in the neuron of your virus, a neuron of your cell, starts replicating. And that's when you get shingles. And it's extremely painful because it's growing in the neuron. And if that neuron happens to be in the eye, you'll go blind. You don't know where the virus is.
Wow.
It is in some neuron of your body.
Wow.
So it's not a different virus. It's the same virus that you got as a child. So everyone who's been infected with chickenpox has a very there's a very, not high, but there's a likelihood that you will get shingles.
So
for which there is now, for which there is so there is a vaccine now, called Zostavax, which you can get when you're 40 or 50 years old, which is basically a booster shot of the chickenpox vaccine that you get to boost your immunity to the virus, so that you will never get shingles. That vaccine is already developed. It's called Zostavax.
Wow. Is there a reason why 40 to 50-year-olds should take that vaccine, not younger? Because I'm just thinking, I have got chickenpox earlier as a baby.
No, I think the thought process is that the immune response to varicella lasts about 30 to 40 years. I mean, if you want to take it earlier, that's fine too. I mean, you'll have to talk to the doctor and figure that out. But I would say 40 to 50 years, it's been tested in 50 years and above in the clinical trials. I don't think it's been tested a lot in the younger people. So, because you can't give a vaccine to a population of people you've not tested the vaccine in.
Right.
So you have to do clinical trials, you have to demonstrate that the vaccine is safe and doesn't cause any side effects, and then it'll be approved. So right now it's approved for the higher risk people who are a little older. But I think over time it might be available for giving to younger people also. I mean, many reasons of why the virus, why chickenpox reactivates, and one of the most common reasons that I've heard about is stress. Stress reactivates the.
Oh, interesting. Because it lowers your body's immune response.
Exactly.
Wow. So I think that's about it actually from the questions that we've had. So I think I just want to thank you again, Naren. This was a fantastic session. So initially we actually planned it. We do internal learn and share every Friday, which is some activity that we do internally. So we initially thought, it'd be great if you can just talk to our team members. And you're thinking it's such a good opportunity to share your knowledge, and you're so passionate about it. So we thought it'd be great if we can open it up to a wider audience. And I think everyone has really loved your session today. So thank you so much.